conservation of energy. We're not making it, we're just altering the form that it comes in. So we're going to get the energy of sunlight in the form of called photons. And we know that light comes in various wavelengths of energy. And plants will use the red and the blues most efficiently. And they're not using the green ones. So the green one actually just bounces right off the chloroplasts, which is why we see them as green and why we see leaves as green. When you talk about photosynthesis, you want to think about where we are structurally in the guts of a plant. We are in the chloroplast. Remember, it's a double membrane organelle, and then it has an inner set of stacked membrane-like structures called thylakoids. So we're actually looking at what happens in the membrane of a thylakoid. Right? This is a phospholipid bilayer structure. There's lots of enzymes in there. That's what we're kind of microscopically thinking about as we talk about photosynthesis. The first big gigantic idea is that in photosynthesis, we are going to take the energy of light and convert it into chemical energy. This is just radiation in a sense, UVA, UVB kind of radiation waves. We convert that energy into a more usable chemical energy. Well, how do we do that? Two-step process. One is the photo, the other one is the synthesis part. In the photo part, we're taking the energy of light, it's hitting the membrane of the thylakoid and exciting electrons, which you can see on the next image for the moment, but they don't have this nice equation down below. So think about electrons, they're just little packages of energy. So when the light strikes the membrane, it excites an electron, it gives it energy, like a power boost, and we're going to use that power in that electron to make ATP. If I kept doing that, I'm going to run out, as a membrane, I'm going to run out of electrons. So I need to replace what I used. Water is going to come in to the cell. We're going to shatter it into 3 parts, 2 separate hydrogens and an oxygen, because I'm actually ripping the electrons off of their covalent bonds and using them to replace the ones that I energized to make ATP with. And when you do that, you get H+, and an O. You do another water molecule, you get another H+, another H+, another O. Those 2 O's bind together, and you make oxygen. The next part then is the synthesis phase, still happening in our chloroplasts. We're going to take the energy of the ATP that we made, which is a chemical component. And we're going to take the energy of those covalent bonds that bind the phosphates together, right? So when you're looking at sort of this idea of an ATP, which is adenine bound to 3 phosphates, right? We first start off with ADP, and we stick another phosphate on to the end. Well, that's where the energy electron came in handy. I use that energy to shove that onto my ADP molecule. Now I've got ATP. These are covalent bonds. There's also lots of other covalent bonds in there. But I'm going to now use this energy from the ATP to staple, to make covalent bonds with 6 separate CO2 molecules. So when I use up all that energy, I end up converting the chemical energy of ATP into a stored chemical energy called glucose. Keep in mind that in every chemical bond, there's energy in there. And so when you digest glucose, you're breaking the chemical bonds and getting the energy from that in a sense. So far, okay? All right, so what's the old, yeah?
I just have one question. So you said to replace the electrons that are excited in the thylakoids, it takes an electron from the water molecule.
Take several electrons from water molecules, yeah.
So how can it just take an electron from water molecule without changing the structure of that atom, like the separate atoms like hydrogen and oxygen?
Well, remember that if we're looking at the idea of a hydrogen, I'm understanding your question, let's just look at this like here. This represents what? Good review question for the exam. Covalent bond. What is a covalent bond really? Sharing electrons, right? So if I have 2 electrons that are here that are being shared by these guys, and I break this bond, then all I have is a hydrogen with no electron, hence a proton. I do the same thing over here. So then I take those two electrons and I shatter the bond such that this doesn't get an electron. So now what I have are two H pluses. And what I have now is an oxygen with a negative overall charge. And is that going where
Yeah,
you're...
I thought that when the bonds broke, that the electrons would go back to their
Right,
respective...
not in this case. It's the way the actual enzyme that's in there. So let me say this. I'm giving us the, well, we'll just do it. Well, no, it's not freshman. We'll do it senior high school biology explanation. Now, freshman college biology, which you get in Bio 211, you're going to spend about 3 days talking about this. There's lots of little enzymes involved in the process. And these enzymes are helping to have worked in such a way that when hydrogen gets ripped off of its electrons, it doesn't actually get any of them. They have a positive force. So, big step is that overall, right, we're taking what's called inputs in a sense. The energy of light, which is radiation energy, right, and with some water so I can get electrons, and carbon dioxide so I can make my sugar, right? The end result is plants produce oxygen and they produce glucose, which is something but chemical stored energy.
Do they use the glucose?
Some plant, yes, they can. So when you look at overall plant physiology, plants in the daytime produce O2 and use up CO2. At night, it's called night cam. They do just the reverse. They can use their oxygen and produce CO2 and use some of their sugars for metabolic energy. So they go both ways. Question?
No.
Oh, okay. That's all right. A lot of students just kind of like, I'm tired of holding my hand up. We'll wait for John. So, okay.
So before we stripped the water of its electron to replace from when we took an electron earlier in the process, we had ADP, and then once we replace the electron, that's when we make the ATP.
In fact, that's what you're using the electron energy for, is to shove a phosphate on an ADP molecule. Yes. Okay. Now, look at the end result. Right? We're making O2, right, and glucose. Well, animals are going to do the same thing, but in reverse in a sense, to a certain degree. When you eat food, whether it's plants or animals, think about it in terms of chemistry. The covalent bonds in the food you're eating, we're going to use those electrons to make lots of ATP, right? Adenine triphosphate. And we're going to use that, what's called the terminal phosphate, the high energy phosphate. We're going to rip it off, and the electrons with that will help me do whatever I need to do in my cell's activity. In order to make ATP, we have two different major schools of thought in making ATP. Keep in mind, it's going to be one of your challenges as an upcoming thriving biology student, is that there's a lot of ways to organize the information and how you need to think about it and talk about it. So if I were to say, what are the two major ways that we can produce ATP? Think, well, there's an aerobic way and there's an anaerobic way. What does it mean to be anaerobic?
To not make
It does not need
energy?
oxygen to make ATP, right? Aerobic means it requires, it needs. Not that it's helpful, but you must, no questions asked, have oxygen to make ATP if you use this biochemical pathway, this process. Well, when we look at the main thing in us that makes lots and lots of ATP, it's an organelle called the mitochondria. And mitochondria, again, this comes down to how you need to think about the question. Mitochondria must have oxygen to make ATP. That is a no argument, no bartering part of its physiology. And what do they produce? Well, they produce carbon dioxide, water, and lots of ATP. We looked at human muscle cells having lots of mitochondria. That means muscles, when they work, right, produce lots of ATP. And cardiac muscle cells have even more mitochondria, which means they're making even more ATP than skeletal cells are. Because right now, a lot of our skeletal muscles are not working, but your heart is always working. It's never taken a break since you were 3 weeks old embryonically. It started to tick and contract. It was what? Cardiac muscle cells have lots of ATP. Therefore, they produce carbon dioxide, water, and ATP. You have a slide that actually shows you that. In case you forget to go back to the last slide, if you look right here, where'd it go? Where'd my, there we go, right there. All right, this shows what mitochondria produce. So how do they make their make sure I didn't skip a slide, hang on there. Okay, so how do they do this? Well, we're going to take glucose, maybe the one that came from photosynthesis. We're going to take the energy in those covalent bonds, along with oxygen, and we're going to rearrange the energy into a more usable currency or form called ATP, adenosine triphosphate. And when we make this ATP, we have waste products or outputs called carbon dioxide and water. That means that this carbon dioxide is the carbon dioxide that you exhale every time you breathe. Well, how do they do it? Well, we need to have oxygen come into the cell, into a cell, and glucose will come into a cell. That part's okay with our little picture here. The fact that oxygen goes into the mitochondria is okay as well. What's wrong with this is that glucose does not go into the mitochondria. Keep in mind that glucose is not used by mitochondria. But we can see it's producing CO2, making lots and lots of ATP. It's also creating water. So what is actually happening here that allows us to make our ATP? That's the first part of it, yes. All right. So let's get to those stages here. So here's glucose, right, in the cytoplasm of a regular cell. All right. So one way to maybe kind of help you think about that, to visualize it, is you would draw a really big cell. Ooh, is that going to show up? Not very well. Let's try that again. That's darker. Okay. Then we're going to draw a nucleus over here. We'll put DNA in it so we know what the heck it is we're looking at. Then we'll have a mitochondria, an organelle, inside of our cell. We'll give it its double membrane, kind of like this. And then we can say that glycolysis, it almost looks black. Let's not do that. There we go. It's happening in the cytoplasm. So this then glycolysis in cytoplasm. So what is glycolysis? It is the conversion of glucose into two pyruvates. It is an anaerobic process. It never requires oxygen. And when you get into Bio 241 and learn about how skeletal muscle cells make ATP, that's going to be a really cool trick.
I just had a quick question. So glycolysis occurs in the cytosol, or the cytoplasm?
Of a regular cell, right?
Yeah.
Is it literally, like, is it like inside a lysosome, or is it just?
Nope, it's
by
just free
itself?
floating in the liquidy stuff of the cell.
Well, I thought the role of the lysosome was to carry nutrients from like food, like glycose, to like transport it to and like break it
It
down.
can, yes, that's one of their
Right.
jobs.
But
They
this
also have
doesn't
this glucose
occur.
floating around.
Okay.
So when you look at this idea here, that means if we are the inside of a cell, that means I must have floating around all these different enzymes, and I'll have all these, what we call it species, all these different things are also floating around. And when they bump into the proper enzyme, they get converted to the very next step.
And the enzyme is not in a lysosome.
Exactly.
It's just
All right. Now, there can be lysosomes that have these, so they can do this. But that means I have to then break the lysosome open to get the ATP out of it that I want. So you also have it just happening for free, in a sense, out in the cytoplasm. Kind of unrestricted. Free range glucose, if you want, rather than being penned up. So let's get to that here then. Glycolysis happens in the cytoplasm of a cell. We know that it is anaerobic. And again, it's going to come down to how we need to think about the information, or what John asks. If I ask you how many total glucose, how many total ATP come from glycolysis, it's 4 total. But how many are usable, or what are the net numbers, right? That's going to be 2, okay? Then we're going to look at the citric acid cycle. These 2 will happen in the mitochondria.
In the mitochondria.
We're going to see that citric acid cycle produces 2, and only 2, ATP. So we'll come back to the bottom one. All right, so let's look at a picture that kind of shows this. Oh, go backwards. Here we go. So overall, we're looking at 3 major processes happening to make ATP. In the cytoplasm of a cell, we have glycolysis, which takes a 6-carbon glucose and converts it, breaks it down into 2 pyruvates, each being 3 carbons. Now, let's go back to the question of what mitochondria need and what can mitochondria use. We know they can use pyruvate because that's what the diagram shows us. This is the more common thing that mitochondria will use. Remember, it came from glucose, which is a carbohydrate. It's a monosaccharide. How many of you are into or have friends that are in Ironman contests, run marathons, or do something called carbohydrate loading, right? Well, if glucose is a carbohydrate, what they're doing is trying to store as much as their cells will hold. Because now you can see why. We're going to take glucose, go through glycolysis, and take the products of glycolysis and let the mitochondria use them to eventually make a ton of ATP. So as long as you've got lots and lots of starch in your system, usable starch, not cellulose, then this can run until we run totally out. Well, what does happen if we run out of these carbohydrates called glucose? You have 2 other forms of energy that mitochondria could tap into. Or another way of saying that, 2 other forms of substrates or fuel sources. And that other one is going to be lipids. That's the next one. Think about the structure of a lipid. You have a glycerol molecule, and then we have these fatty acids that come off of it. And if I have 3 fatty acids, what have I formed? Triglyceride. So let's draw some fatty acid chains. Remember, each one of these is a unwritten carbon, then they have hydrogens coming off of them. And we just have shorthand to show this. Well, there's a process where you're going to take 2 carbons at a time and you break them off. That would give you CH2, CH3. And there's a process in your cells that can actually use this to make ATP. And it's called beta oxidation. And you're not responsible for knowing the idea of beta oxidation. It's just to give you an example of how we can use lipids for more ATP production. And then you keep chomping these off like 2 at a time. So the cool thing about this is when you learn this in 211 and other physiology classes is that the bigger my fatty acid is, the more energy it has for us. Glucose is set. I'm only going to get 2 net usable from glycolysis. I'm only going to get 2 from the Krebs cycle, and around 34, 36 from the electron transport chain. I can get more than that if I burn fats. If it's a really nice big long fat. Well, in a sense, that's why we have this, right? This is not to keep you warm. It is not to pad you from bumping into people in the elevator. This is stored energy. What you want to think about is we're spoiled today compared to what we were in terms of our ancestors. Let's go back 20,000 years ago. How many Safeway stores were there? None. Okay. Maybe 1 or 2 beginning to show up. A Starbucks, maybe 1 on every continent, right? But the idea was that we were nomadic organisms, animals, because we're still an animal, and we did 1 of 2 things. We either ate as we were migrating to better weather and better resources, and we nibbled here and nibbled there and nibbled here, and you came across a big abundance, like maybe a whole field full of blueberries, right? You didn't eat just 2 or 3 and put a few in your pocket, if you had a pocket. You sat down and you gorged yourself silly. You came across a dead animal, it might have been dead for 2, 3, 4, 5, 6, 7, 8, 9 days. You consume as much as you possibly can. It's feast or famine. There'll be days that you won't even be eating. None. And days where you're going to stuff yourself silly. So I asked my animal behavior students this when we have the class. How many of you like all-you-can-eat buffets? All right. Now the question I always ask my animal behavior students is, why do you like anything? I mean, why do you like the smell of babies? Why do you like sex? Why do you like to eat a lot? I mean, there has to be a biological reason for that like, and that is that you do more of it. So you like to eat because it allows you to store calories. Now you can go for a couple of days without having to eat. To give you a really good example of this, there's a guy, I mean, he's not really crazy, I think he's kind of crazy, who lives in England. And if you saw him walking out of a pub in London, you would think, dude, okay, you've had a few too many Guinnesses and way too many bangers and mash. That's hot dogs and potatoes. He has about a nice big 14, 15 pound layer of fat in his belly. He kind of jiggles as he walks. Right? Now, you're going to see him, well, this is a really ugly picture, but he wears a really tiny Speedo. So you're going to see him in a Speedo, and he's going to wade into the English Channel, and he's going to swim for the next 30 hours, non-stop, to the shores of France. And when he gets out, he's lost 17 pounds. That's why he has the belly. He likes to swim, it's crazy, the English Channel. But also let's realize that fat is an energy reserve, and he's using that all the time as he is moving. Well, that was sort of what got my curiosity up. My work in graduate school was to understand how animals know to convert from using lipids to the next possible available fuel source, which might be what? Proteins. And how are proteins stored in us? What's our biggest source of proteins? Muscle tissue. Well, that's a good and the bad. If I'm a migratory bird, which is what I studied white-crowned sparrows. If the breast meat, which is their flight muscles, are what powers them to fly, it's also the first muscles they begin to digest to get energy. So it's sort of like dissolving your own engine. So the question I was asking is, what is it that tells their metabolism, their physiology, that, dude, we are totally tapped out of fat. We need to start burning something else. And it's a hormone called corticosterone. What you look at is when birds fly these long distances. Some birds will fly 6,000 to 7,000 kilometers non-stop. They don't eat, they don't drink. But when they do, it's called put down, they'll have a mid-resting point, and they might be there for 2 or 3 months, stuffing their little faces as much as they can to regain their fat. When you capture them when they first land, if you can capture a little white sparrow, they're cute, hold them like this, their little heads are peeking out here, little feet back here, don't squeeze them, right, because they squeeze pretty easily. But if you hold them and blow on them, you can blow their feathers away off to the side. You can see through their transparent skin. You can see their internal organs. You can see they have no fat at all. None. Zip, zilch. So the question is, then what are they using for fuel? And we realize they're actually metabolizing their own skeletal tissues. You will do the same thing. If you have totally run out of fat, you've got zip, none, zilch, then you're going to start metabolizing your own skeletal muscle tissue. It's not a good thing, but I guess you can take a bad thing and turn it into a usable thing. Look at photographs of World War II survivors, all right? Auschwitz, Dachau. Those people that were kept in prisoners of war concentration camps. Look how skinny they were, right? They were basically skin on skeletal systems because they were metabolizing their own fuel source of muscle tissue. How many of you saw the movie that came out this last summer of Heart of the Sea? About the whaling crew? All right, if you haven't seen it, when school is over, watch it. or read the book Heart of the Sea. Heart of the Sea is about a whaling ship in the 1800s that got smashed by a large male sperm whale. And the crew had to abandon ship. And they had to abandon themselves into these little whale hunting rowboats. And they were adrift for about 6 to 8 months out in the ocean. All right? And remember, back in those days, they didn't have outboard motors. So if they came across an island, they would maybe get a chance for some water and such. Long story short, end of the whole trip, there were 2 boats that were finally found adrift out in the ocean. And there were 2 survivors in each of those 2 boats. In one boat was the captain and another crew member. And the other boat was the first mate and another crew member. And they were found clutching skeletal bones in their hands, arms and leg bones of their crewmates, who they had eaten. Because the crewmate said, I'm about to die, dude. You can either toss me off and feed me the fish, or you can eat me and get some energy out of me. And that's what they did. It wasn't like they were killing you so I could eat. It was more like, you said when I die, eat me, so you have a better chance of surviving. An author in Nantucket bumped into the captain a year later in a bar, of course, sailors. And he got so enthralled by the story, he wrote a book, Moby Dick. The idea was that the sperm whale had actually attacked the ship because it was one of the very first copper-hulled ships. And the sonar from the sperm whale bounced off the hull, came back to this male sperm whale. And he probably thought, that is one big dude. I have competition now for the girls. And they ran each other for mate selection. He probably thought the ship was a big sperm whale and went to ram it, and unfortunately it wasn't a sperm whale, it was a ship full of people. And that whole story then is to realize that we will metabolize ourselves. You will do this until you lose about 1/3 of your skeletal system in terms of muscle mass. But as you do it, you produce toxins called ketones, which are very deadly. So you're going to die toxicity before you actually run out of usable energy. Because you do have...
So is that what ketosis is?
Yes.
So, but like Inuit tribes where they only have protein and fat for all of their
Right.
lives. They live all their lives pretty much in constant ketosis.
Yes, they do. Because
it's a toxin?
It's a byproduct of what's called deamination, the removal of nitrogen from the amino acid structure.
Okay.
Yeah.
So, in this case, the toxin won't kill them?
Not, because they also have fats, and they're eating a little bit of plant matter every once in a little bit of blue moon.
Okay.
And they're also healthy. They're not physiologically stressed, which also compounds the problem. And the reason you guys do this is because you have one really selfish organ that always needs a supply of ATP, and that's your brain. Your brain will tell your body to digest itself. Yeah, that's all it has in terms of sources of making ATP. So let's get off the topic here and go back on topic. The idea is that mitochondria need oxygen, but we now know they have access to 3 different types of fuels, right? A carbohydrate known as glucose, but it first must be broken down into pyruvates. We can use fats, as long as they're small chains of carbons. We can use proteins, as long as they're an amino acid part, as long as they're the monomers. Which is the big bottom line is, we're using the monomers only, not the macromolecule. Now, we're going to call the citric acid cycle a mysterious black box. All right? Stuff's going to go in called pyruvate, and out comes 2 ATP. And usable energy in the form of electrons that are bound to an enzyme called NADH. This is called an electron transporter. It's going to shuttle the electrons from this process, like north part of Seattle, to South Seattle, where I can now have another factory that can use those packages of energy. So citric acid cycle happens in the mitochondria. It makes 2 ATP, and it takes electrons from our pyruvates and shuttles them using NADH to another process called the electron transport chain. Now that happens, as you can see by the cartoon, in the inner membrane of the mitochondria. So it happens in the inner membrane. And it's a process called electron transport. So we're transporting electrons. Now, it depends upon what book you have. and it's sometimes frustrating for us as instructors. One textbook used at UW for their biology 180, 200, and 220 series says that you make 29 ATP. I have 2 books on my shelf in my office. One says you make 30, one says you make 32. I have another book that says you make 38.
Yeah,
What's going on
I
here?
seen that last night.
All right, so here's our rule. It kind of depends upon the animals they're studying, how they did the study, because if we looked at all your results of amylase from yesterday, even though all of you did maybe the same salt experiment, you got different results. For us, let's just do this. Remember, quite honestly, what is this class about? A, to teach you the basics of biology, give you a prerequisite for Bio 241 if that's where you're going, but it is also, understandably, honestly, a weed-out class, right? To see if you can memorize things we ask you to memorize. So this will be our Bible. This is what you need to know. Regardless of whatever other book you've got, if it goes against these numbers, ignore them, and we're going to focus on these. We can all be consistent then, right? We make a total of 4 from glycolysis. We actually have 2 usable ones. We only make a total of 2 in the citric acid cycle, otherwise known as the Krebs cycle. We're going to make 36 from the electron transport chain. And this is the part that requires, that needs to have oxygen present. So now if I said how many ATP can come from mitochondria, then what would the number be?
From the mitochondria,
Uh-huh. You get 38, right? Because 36 plus 2, right? Because this happens in the mitochondria, right? If I were to say, starting with a single glucose molecule, how many ATP could we possibly make? What's the maximum number? Then you go, we got 4 up here, 2 here are 6, and 6 and 32 are 42, right? So you got 4 and 2 are 6, and 6 more are a total of 42 from one glucose molecule.
But isn't 2 of them not usable?
I said total.
Oh.
And see, now what if I said how many usable ATP, then it's 2 less than that. So again, it comes down to how you need to remember the information and organize it in your brain. Because if you were, let's say, in a laboratory and your graduate professor said, well, how many ATP were actually completely produced
4.
versus how many can we actually use for the next cellular process. So it is important how you keep these guys in mind.
4 is
Right.
made, 2 is
If you look at this part of your slide, we realize that this is why sometimes you got to be careful of what you read on the internet. This is the only ATP produced, far less than produced during aerobic respiration. They're pointing to that, and that came from what process? Glycolysis.
yeah.
Are 2 ATP really the only ATP produced?
No.
You make 4, right? So you can't trust the internet. It depends on how you interpret the information. But what we can see is that, and what we're trying to talk about here is, if we cannot use the pyruvates, if you're low on oxygen overall, then we're going to convert the pyruvates into a different thing, something called lactic acid, or lactate. That will then leave the cell. And it can be used by your liver for other stuff. Bacteria do the same thing. They make pyruvate. But when they can't use it, they convert it into something way more fun. Ethanol, which we could drink and have fun with. So if only our skeletal muscles could convert pyruvate into ethanol, you'd all be exercising all the time, right? If you wanted to get a buzz. But it's what happens when things cannot be used up quick enough. They have to be altered and gotten rid of. Because another name for pyruvate is pyruvic acid.
Yeah.
And do acids affect pHs? Do pHs affect enzymes? Right? And this is happening in the cytoplasm of your cell. We have all those enzymes for glycolysis. You don't want to damage them, otherwise you couldn't run glycolysis. So the cell has to get rid of it. And it first turns it into lactate. That's the fermentation part. And then it goes into the blood. And be used by other organs for other stuff. I want you guys to see what this thing works. We got to add one more word to your understanding. Is that memorize none of the steps in here. It's just an example of enzymes, how important they are. Memorize none of the steps in here. Just more examples of how important enzymes are. Alright? Memorize none of this. This is the electron transport chain. But here's what I do want you to get from this. The whole idea is I need to transfer electrons from thing to thing in the membrane. They're enzymes. And as I transfer my electrons from thing to thing in the membrane, I allow hydrogens to leave the inside of the mitochondria's membrane to go out here. I want lots of hydrogens out here. When you try thinking about lots of something in one place and very few of them in another place, you're thinking about diffusion, concentration gradients.
They go outside the cell membrane or the membrane.
Oh, they go outside the inner membrane. This is the inside of the inner membrane. This is the outside of the inner membrane. We can see then that those hydrogens are going to diffuse back into the cell. This thing right here we need to know. This is what's called your ATP ACE pump. It makes ATP. It's also known as ATP synthase. Synthesize. ACE for enzymes. I want you to see how this thing works. So that's why it's called the electron transport. We're transporting electrons. That causes hydrogens to be stuck outside. All these different things you see here are the names of each of the different types of enzymes. Don't worry about them. We're just going to call them things. But I want lots of hydrogens out here. And the hydrogens are going to come in here. And this thing is going to spin like a windmill, like a motor. And it's going to create ATPs. And in the 6 minutes I'm going to take to do this, I want you guys to see how this actually looks and works. Let's go to these guys.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase,
Don't want the names.
cytochrome BC1, cytochrome oxidase,
ATP, same.
What
Nice.
mitochondria you use to make ATP?
I'm sorry, well, yeah, what are you use to make ATP? They
In
do it the
addition to
following
these
way.
Makes
complexes,
ATP
2 mobile
in the
care nets,
mitochondria.
which the other 4 hydrogen ions are pumped across the membrane,
Let's go back
hydrogen
to-
ions. The 4 electrons
Gotta find
to
it. There
make
we go.
ATP
All right. So all these little pink things are hydrogen ions. They're going to diffuse down through the pump. You can watch the top part of the pump move.
ADP, and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation.
The bottom line is basically this. As hydrogens come into the top, cause the top part to rotate. As the top part rotates, it causes these things like those little paper games with your fingers used to play in elementary school to open and close. That allows an ADP to come in, and I can turn into ATP. This part rotates, this part opens and closes. Well, I want to show you a real version of this. And this was done with a lot of studies about molecular structure, and it's upside down now compared to the things we just saw. But I want you to see how fast this thing works. This thing makes 100 ATP per second. And you have thousands of them in one membrane of one mitochondria. And you have millions of mitochondria, which means you make a ton of ATP per second. The cool thing is it looks like a little biological motor. And it's spinning, 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 spinning.
Based on an incredible series of scientific discoveries, only the colours show artistic license. ATP, or adenosine triphosphate, is the energy currency of the cell. ATP is produced by a tiny molecular rotary motor, rotating at up to 7,000 RPM. These are so small that 100,000 would fit side by side in a millimetre. A current of protons drives the motor, unlike man-made electric motors which use electrons. This portion of the enzyme is where adenosine diphosphate is combined with a phosphate ion in the presence of a catalyst to produce ATP, which is then released, making way for the next cycle. A top view of the enzyme shows the sequential operation. Almost every biochemical process in your body requires ATP.
Every different color thing is a different protein. If I had more time, because I'm out of time, this is an argument for creation. This was called irreducible complexity. That you can't reduce this anymore without breaking it, it no longer works. That this could not have evolved from anything other than an intelligent designer. This link would show you the structure of a bacteria flagellum. It looks an awful lot like this, but it doesn't do the exact same thing. If you were to modify that bacterial flagellum, we'll show you this in the next class period, that you could actually end up creating this. So it is possible. We have now begun to find ways to prove that it could have evolved from a different structure. So be on time tomorrow in class, folks, so that you have all the time you need to take the exam. You're done for the day.
Oh, yeah, I skipped that one too. Like I
No, you went over briefly. Flagella what?
I get what it
Flagella is like a tail, it's a movement protein or something, and it spins.
Good question. So see why
Yeah, I don't know if it's a movement protein, but
Sinase itself. No, this sinase is CY. This is sin. Sin.
it
Oh,
enables like bacteria to move.
there we go. I'm sorry.
See,
Did you go over the function of how the...
Synthase. I'll get the pH. I'll send you guys an email to fix that. Go over what? Yeah. We did that probably early last week. We talked about cilia, flagella, and the 9:2 ratio of the microtubules inside. Then I showed you guys the Oompa Loompa, the lower protein that walks along the microtubule. Okay. Yes, it should. It probably is.
Oh, and it's still